Thank you. Let me start out by, Let me start out by explaining what I actually mean by intron retention. In metazoans, most splicing happens co-transcriptionally. So, is there a pointer?
Ronnie, did you take the...
Oh, this is it. Sorry. Yes. Thanks. Yeah. So most splicing happens co-transcriptionally. And so while RNA polymerase II is transcribing this gene here, where the boxes indicate exons and the lines indicate introns, nascent RNA emerges and is already being recognized by splicing factors. In fact, most introns are being efficiently spliced either already before or shortly after cleavage and polyadenylation. But some introns, like this orange one here, are not effectively recognized and remain in the RNA. So what happens to these transcripts containing retained introns? Well, very often export from the nucleus to the cytoplasm is very inefficient and they become degraded by the exosome. Transcripts that do make it into the cytoplasm very often contain premature termination codons, and that is a trigger for the nonsense-mediated decay machinery, so they become degraded too. And there is only a minor fraction that do not disrupt the open reading frame and actually have the potential to give rise to a variant polypeptide. Now, in mammals, it has been thought that intron retention as a phenomenon is very rare. And this is in opposition to organisms like plants and many protists, where it's actually the most prevalent form of alternative splicing. However, in the last couple of years, a few examples have been known in which cells use intron retention as a means to regulate a gene expression program. For example, there is a group of genes that are important in synapse function that contain introns which are retained specifically in non-neuronal cells, and there they mediate impaired nuclear export and exosomal degradation. And in another example, a different set of genes contains introns that become progressively retained during granulocyte differentiation, and in this case, degradation is mediated by NMD. So examples like this have made us interested in conducting a comprehensive survey of the rate and also the regulation of intron retention in mammals as a whole. And by doing this, we hope to maybe find more biological functions for this phenomenon. So this is exactly what we did. We assembled a broad collection of RNA-seq data, both from the literature and some from our own lab, comprising 52 human and 65 samples from mouse, and covering a broad spectrum of different tissues and cell lines, both transformed and non-transformed. And what we did chiefly was to count the number of RNA-seq reads that support retention of an intron, as well as the ones that support splicing, and calculate what we call the percent intron retention, or PIR. And then, of course, we also introduced a number of quality control and filtering steps, and we conducted quite extensive validation by RT-PCR. So when we look at different thresholds of PIR in human cells, then we see overall that between around about half and at least 9 to 10% of all introns are retained at least in one situation. On a gene level, this translates to between around 40 and around 75% of all genes which have at least one intron that is retained at least sometimes. We have strikingly similar results also for mouse. And I should explain that in all these graphs, the solid blocks represent what we actually measure in the samples that we have, and the hatched part represents an estimate that we made by extrapolation. So intron retention seems to be not too uncommon in mammals overall. But now let me jump right to a key observation that we made, which is that intron retention seems to be very strongly linked, or at least to quite a strong extent, to gene expression. So for this analysis, we grouped all the genes in every sample into 10 bins, ranging from low expression to high, and scored the percentage that were retained by at least 10 PIR. And quite obviously, you can see that there is an inverse relationship between the extent of expression and intron retention. This is again true both for human and for mouse. Now, I don't have time to show you that, but we also demonstrated that both impaired nuclear export as well as NMD play at least some role in regulating the steady state levels of at least many of those transcripts. What I do want to show you, however, is one example where we think this plays a role. We analyzed data from a time course experiment where mouse ES cells were differentiated into glutamatergic neurons over 6 time points. And shown in this graph is the degree of intron retention for all those introns that change by at least 15 PIR. But obviously, the largest group of intron gains intron retention, and their expression of the genes that they're contained in is higher in ES cells and it goes down in neurons. On the other hand, for genes that are downregulated by at least 5-fold, introns that they contain become more and more retained over time. We also wanted to know what the functions of the genes in both these categories were. And for the group of genes with increasing retention, we found a broad spectrum of associated gene ontology terms, including also terms related to proliferation and DNA repair. And we also noticed that there are a number of genes with very specific functions in only some cell types or in specific situations. For example, and this is RT-PCR validation, we found one that is a synaptonemal complex member in meiosis. This is called epsilon, which is a DNA polymerase that is important in DNA repair. And this gene here is actually expressed in mature neurons, but in a different type, so in serotonergic and not glutamatergic neurons. In all cases, the mature mRNA is downregulated and intron retention goes up. Now, for the group of genes with decreasing intron retention, they're associated only with 2 very specific terms, neurotransmitter transport and synapse, both of which are actually are obviously important for mature neurons. So we also wanted to see whether this what we observed here holds true on a larger level. So we grouped all of our samples into neural and non-neural samples, as well as muscle and non-muscle samples, and ES cells versus non-ES cell samples. And for each grouping, we classified the genes into 3 categories: ones that were relatively low expressed, or average, or overexpressed with respect to all the other samples. And then we scored the degree of intron retention. And indeed, the class of genes that are relatively poorly expressed have the highest level on average of intron retention, and the genes with relatively high expression, which are also the ones that are most frequently associated with Go categories that are relevant to the tissue group in question, are the ones that have the lowest degree of intron retention. So a picture emerges in which intron retention mostly affects genes when they're low expressed and maybe out of context of when their function might even interfere with the cell type. So I've shown you these 2 important points already, and then I didn't have time, but if you're interested, you can have a look in our paper. We also have data to support that intron retention is coupled to slow elongation and also to impaired recruitment of splicing factors. And together, that sort of is compatible with our favorite cartoon model, in which transcripts from highly expressed genes are spliced efficiently, but ones from genes that are not really highly expressed and induced very lowly also have suboptimal recruitment of splicing factors, and they have a higher frequency of intron retention, which then leads to elimination of those retained intron transcripts. So altogether, this has the potential, we think, to function as an extra filter against inappropriate gene expression. With that, I want to acknowledge the people involved in the lab of Benjamin Blanco. These were Manuel Erimia, whom many of you know because he's now here at the CRG, Nuno Barbosa Maraish and Sandy Pan on the computational side, as well as Emil Nackman and Thomas Gonatopoulos on the experimental side. And we had help from our colleagues in Brendan Frey's lab at the computer engineering department, as well as Eduardo Eras from UPF. And with that, I thank you for your attention.